everyone, and thank you for attending today's webinar, The Next Wave of AI, the Impact of LQMs on the Pharmaceutical Industry, presented by Fierce Pharma and SandboxAQ. I'm Rebecca Kushmiter, and I'll be your host for today's webinar. I'd like to introduce today's speakers. We're going to be joined by Joseph Lehar, an advisor to SandboxAQ, and Andrea Bortolato, VP of Drug Discovery at SandboxAQ. You can find their full bios in the Speaker Bios panel. All right, now let's go ahead and take it away, gentlemen.
So first I just wanted to give you a quick view of the agenda. We're basically going to be talking a little bit about AI and drug discovery. It should be review for most of you, but for those who aren't, it might be helpful. And after that, Andrea is going to do the real meat of the presentation, describing more about what's happening with large QMs. And basically, I'll mention a few comments on the impact on drug pipelines, and then we'll have some questions afterwards. So first, just as a broad introduction, as many of you I'm sure are aware, artificial intelligence is becoming much more of a feature in almost everything that affects our lives. So this graphic here just sort of highlights how it affects many different stages of life, where essentially coming up with novel ways of enabling fertility treatments for young children. There are an increasing number of gene therapies that can be both identified and addressed, where artificial intelligence is helping with that. Well, on the topic of designing drugs for diseases and different conditions, this has been an intense focus of area for AI over the last couple of decades. And we're also moving additionally into using the harvesting of large amounts of information on patients to help manage lifelong patient journeys, or even try to coordinate healthcare resources. There's a large real-world evidence industry developing in many different sectors around that. And even, just to complete the picture, increasingly AI is being used to manage the quality of life until death. So on to the next slide. If we focus in on the drug discovery process, one way to think about how AI is impacting is to break down drug discovery into the different stages of work that are required. So the sort of classical way of viewing the drug discovery process is really highlighted by the arrow in the center of the picture, going through the various different stages. And I've augmented this by basically putting on what happens before and what happens afterwards. So before you start with the process, you first have to decide what disease you're going after. And that basically involves looking at an indication, which might be something as vague as breast cancer, and dividing them into distinct groups that have different biologies. And artificial intelligence is using patient variables, imaging, and molecular profiles to do a lot of that classification into sub-diseases very effectively. The next step is really, once you've got a disease that you want to go after, it's useful to identify a target. What is it that you would want to modulate to improve patients who have that condition? And there, the AI techniques have been applied to literature, to molecular profiles, and to phenotypic screens to, again, do a very good job of identifying the targets and assisting with the validation of those targets through experimental procedures. Once you've got your target, you then have to understand how might you modulate that target. And that's typically done with a molecule, where essentially you'll try to put a small molecule or some biologic onto that target and thereby modulate the behavior. And there's a larger area of research that's been going on for decades now, where you can optimize chemical structures, or you can look at run ligand screens, or you can do high-dimensional phenotypic screens to basically first identify which molecular lead is going to do well against that target, and then also further optimizing that molecular lead to enable you to actually make a workable drug out of it. Having got to the point of an optimized lead, you then need to go through development. And development has two major phases. The first phase is preclinical development, where you basically test out in models of increasing complexity, starting often with things as simple as cell line or tissue assays, and ending up with animal studies to confirm safety and efficacy. And once you've convinced yourself that your drug is going to work well, you can then take it to humans, and then the clinical process begins. There are 3 phases to clinical trials, as many of you will know. Phase 1 is aimed primarily at toxicity and safety. Phase 2 is aimed at efficacy, and Phase 3 is aimed at large populations, making sure that nothing untoward happens in large groups, and that you have a good effect through the whole population. Once you've got through all those phases, you typically have a drug, and that drug can then be approved and brought to the market, at which point AI doesn't stop, because there's essentially a lot of optimization that needs to be done on how to manufacture the compound, how to deliver it to your market, monitor the safety of it, and possibly consider repurposing the drug. And again, there's a lot of work in artificial intelligence being applied in that sphere as well. And so to dive more deeply into LQMs, we can then hand off to Andrea.
Thank you, Joseph. So large quantitative models can be considered like the next frontier of the evolution of the synergy between computational chemistry and AI. And LQMs are really powered by the transformative deep impact of machine learning solution combined with state-of-the-art physics-based simulation. So like in this slide, I think I'm looking back, and it's interesting to look at the history of rational drug discovery in pharma and biotech. This timeline kind of showed the adoption of innovative in silico technologies in the private sector. And the history of drug discovery has been really in the hands, right, from the very beginning, in the hands of incredible scientists, medicinal chemists in particular, that have been able to design and evaluate in their mind new ideas, new molecules. They've been the real driving force of drug discovery. And so rational drug discovery has always been happening, if you want, in their mind. And they were predicting the result of molecular changes based on the structure-activity relationship, understanding, based on all the data, based on their experience. I think in the 80s, and from the 80s to the early 2000s, math, statistics, chemical, physical properties started to help more and more medicinal chemists try to provide, if you want, a deeper understanding of how the molecular structure was related to the biological activity. And this has been, I think, potentially the beginning of impact of computational chemistry in drug discovery. But then there was a key important step that was the beginning of the GPUs that were initially used for molecular dynamics. So this changed the ability of structure-based drug discovery to be more impactful and valuable. It was a slow process, and I joined this journey at that time, and it was a very challenging start, right? The methods were quite simple. The impact was limited. I would say probably it was mainly used in a retrospective fashion. It was like an hypothesis generator tool, if you want. So if you work with medicinal chemists from that period in particular, they still tend to look at the impact of computational chemistry as kind of a retrospective value that could add some insight, but it's really, really limited in the way it can be valuable in reality. But the transformation started with the GPUs, and that kind of allowed molecular dynamics to reach timescales that before were just unthinkable. So the molecular dynamics started at that time to create, I would say, a reasonable thermodynamic ensemble of the conformational landscape of the system in study, right? Unlocking quantitative affinity prediction, for example. That was a key initial step. And then in parallel, like GPUs have been also able to transform AI and machine learning. So deep learning solution, they started to be able to memorize, massive amount of data. And this was just the beginning that in the end provide these foundational models. And a clear example is protein structure prediction. So our ability now to predict the 3D conformation of a protein is massively improved. And this is really thanks to the ability to have a GPU-driven AI model able to memorize every single protein structure that could be X-ray or cryo-EM that is available in the protein data bank. So that was really transformative. And I think now with LQM in particular, we're reaching that point of non-return where even medicinal chemists that before were not fully convinced about the value of this solution start to see that as a huge opportunity. I kind of like to call this phase like the clip phase of drug discovery. In the viral life, there is this eclipse phase when the virus infects the cell, but they still kind of wait and getting ready to hijack the cell, really. So it's similar now. I think LQM and AI and simulation are in drug discovery in pharma and biotech, but they are still getting ready. And I think soon we will see a real impact in the way they will transform the way we do drug discovery. And it's an exciting time, really. It's kind of showing the revolution that is going to happen. And the time between innovation and research in academia and implementation in pharma and industry is really changing. Before, it was a big, long wait before really seeing molecular dynamics having impact in industry. But now we see this time frame is start to be extremely short. Everything is moving extremely fast. The technology is evolving and is becoming obsolete extremely quickly. And this is really thanks to groundbreaking AI innovation happening everywhere. And so I feel like for the first time, pharma and biotech feel the pressure, really, the pressure of quick adoption. You need to be agile. You need to bring innovation as fast as you can, because otherwise there is a real risk to be left behind, to lose the race, or to miss kind of amazing opportunities. So in that direction, I think LQM is going to be extremely powerful and valuable. So here I'm kind of providing an example of LQM. So the overall vision is really try to combine building blocks that are based on physics-based methods with AI ML components. Try to bridge the gap also toward the experimental data that can be available. So in this slide, I'm kind of highlighting how you can combine structural information that we discussed before, like taking all the protein data bank data and create this comprehensive data-driven solution to predict ligand protein 3D complexes. And then you can expand that, right? You can move forward, including the dynamics of the system. That is also extremely important because we don't want to have a crystallized vision of the protein. This is very dynamic, and the flexibility is essential to predict the biological outcome. And this is challenging still. Try to go from the static 3D picture to the final ensemble and thermodynamics conformational landscape of the system is crucial. It's crucial to go to the next step, that is really predicting the affinity. And so here is try to go into the quantitative aspect of these large models. So we want to understand the molecular interaction at a level that is enable quantitative prediction and decision-making strategies. And that is really important. And I think here there is an important aspect that is trying to find the best compromise between speed and accuracy to create efficiently a large number of data that could be used as a training set for these kind of universal protein ligand physics-based affinity models. Then the next step is try to include all the experimental data. And we can use in this example a proteochemometric solution that really takes everything available in the literature. It could be patent or even internal data from other projects in your company, and try to bring all that information together. And so this graph neural network at the end will be able to take a molecule, co-fold it in the protein target environment, understanding the dynamics, extrapolate the binding affinity, and then using this kind of PCM approach, link it to the probability of reaching the desired functional activity. So very often the key here is try to ensure you have all the information available when you make a decision, right? So the idea is LQM will provide a tool that gives superpower to an expert medicinal chemist. So when that medicinal chemist is deciding what molecule to make next, they will have in hand all the best prediction from physics-based method combined with all the data available from the community and other data from their company. And that will enable them to go faster, to make fewer and better molecules in each design make test cycle. a molecule with a higher probability of success in the end to reach the clinic. These here are kind of few examples of how we can create specialized LQM platform, from AI and physics-based building blocks. And it's very important to ensure these LQM are optimized for the problem you're trying to solve. It could be the protein target, it could be a modality. The reality in drug discovery is still extremely challenging to solve the problem that we are tackling. And in many cases, you cannot have a general solution that is broadly applicable and always work. It's just too complex, the problem we're trying to solve. So it's important to understand what you can simplify, what is crucial for that question, and really create optimized, specialized, bespoke solution or LQM models. The idea is to try to expand the applicability domain of AI and the training set available with physics-based simulation and maybe Bayesian strategy to try to create the best possible workflow to answer the question you're trying to solve, to move forward and find a clinical candidate. So I have 4 examples here. Like the first one on the top left is a creation of an LQM optimized for neurodegeneration. Is combining a bespoke physics-based docking solution powered by an optimal force field that is really specialized for the chemistry of interest, the molecule of interest. And we combine that with AQFEP, a large-scale absolute free energy perturbation, extremely fast strategy for virtual screening. So in this case, when we applied this approach, this LQM, the hit rate was between 20 to 30 times superior to the IEEE screening that was used originally. And this is on a phenotypic screening definition of success. So it's very challenging and very realistic. And at the same time, we were able to screen a 20 times larger chemical space compared to the HTS. And we tested experimentally hundreds of times fewer molecules. So this is kind of how we can bring value. And this is really, we are obsessed with kind of deep impact at scale in these cases. The second example on the bottom left is another virtual screening strategy, combining our AQFEP solution with active learning and GraphDoc on an oncology and druggable target. And it proved to be extremely effective. We identified several scaffolds that enable the hit to lead and lead optimization phase. But we can go beyond that, right? Like the third example on the top right instead is a different type of strategy where we try to combine the modern proteochemometrics approach that I described before with knowledge graph and physics-based method like AQFEP to create a fast transformative LQM for target identification using reverse screening. And this solution has been able to take advantage of all publicly available data and also internal omics data from the client. And we were able to understand the molecular interaction between the protein, the target, and provide powerful predictive solution that were proven to be correct experimentally. And then the final 4th example on the bottom right is really try to expand the applicability of LQMs toward clinical development. And we're really excited about this direction. We're investing a lot of research. I think this will be the next frontier. So the vision here is really to try to deploy an LQM that can decode patient stratification, patient selection, try to minimize the side effects, try to maximize efficacy, reducing the clinical trial size. And this probably is where there are the biggest challenges. So it's extremely exciting, could be extremely impactful for patients, for a medical need. But there is a lot of important research and development that is needed. And I pass it back to you, Joseph.
Absolutely. So I just finally wanted to comment a little bit on how the LQM approaches can impact the path to a drug. So coming back to the picture that we showed before, the arrows didn't come out very well in this projection anyway, but imagine nice arrows where you see at the top row ending up in a drug. Essentially, if you map out the different stages that it takes to get to a drug, you begin with target discovery, then you have compound discovery, then you have preclinical validation, and then the 3 different clinical phases, right? And as described before. And the thing to keep in mind is that the traditional approach to this, what we've been doing for the last many decades, is actually very slow. It takes about 13 years to go through the whole process, starting with target discovery. It's very costly. It costs almost $1 billion to get from the beginning to the end, and it's quite risky. Even when you start with validated targets, 90% of them fail when you take them through the clinical process, and that's largely due to the fact that we have a very hard time predicting what's going to happen with a molecule as it gets through into relevant systems and translating the models that we use for discovering molecules through to the human system. So if you think about how LQMs can affect all of these, essentially the LQMs will have a very profound impact on the target discovery process in the way that Andrea discussed. There's also a very profound impact on the compound discovery. You can basically optimize the design of the molecule very effectively. Those two optimizations and enhancements will lead to direct consequences on all of the later stages. Conceptually, basically what's going on is if you choose a better compound, you're more likely to succeed as you go through all the different stages of translation and so forth. And also, if you have if you've designed with toxicity in mind, you will have a much lower failure rate on the toxicity and translation efforts as well. And you can also apply AI methodologies to optimize the design of drug screens as well. So when you take all of these components into account, what you can find is that really when you move from the traditional path towards the LQM-driven path, you can reduce costs by quite conservatively about half, so about 50% reduction, increase the probabilities by a significant amount, and reduce the time scale from about 5 years to cutting off 2 years or so. You get similar improvements as you move along the way. The numbers given at the bottom are cumulative. So at the end of the process, you end up with saving about 20% in the cost, saving about 4 years on the discovery and development process, and increasing the certainty of discovery by a factor of 2. And that comes primarily from the fact that you have higher quality molecules that are much more likely to translate in the clinical proof of efficacy. So taking all of those three things into account, you will have a major increase in the net present value of your campaign to identify a drug by factors of 2 or 3. And essentially, it becomes a very sensible choice to apply artificial intelligence and LQMs in particular to enhance your drug discovery process. And with that, I think we move on to the moderated discussion.
Thank you, Joseph.
All right, well, thank you, gentlemen. We will be moving on to our questions now, and I see we have quite a lot of questions in the queue. We'll get to as many of them as we can in the time we have today. So to begin, can you describe how AI LQMs can be deployed in lead optimization and structural activity relationship modeling?
Yeah, thanks. That's a great question. So the key idea, I think I can use this slide actually that may help to kind of provide a little bit of a description of the design make test cycle that we are using driven by LQM. So the vision is try to combine key innovative technology that are transformative in a way that is compatible with the hit to lead and lead optimization, right? And so I think all these kind of pieces are extremely game changing and extremely impactful and valuable, but when combined together in an effective way, could be much more effective and really try to move faster the hit to lead and lead up toward the clinical candidate identification. So we deploy as a first step like a fast chemical space exploration. I will describe and touch a little bit on that. So we can combine a QFEP with active learning. A QFEP is our absolute free energy perturbation solution. We can combine it with active learning because we can profile, one molecule on one T4 GPU in a couple of hours with our absolute FEP approach, but still, it's potentially thousands of molecules we can profile per day, in a normal cloud infrastructure. But it's not enough if you think about the option available. Like there are commercially available, there are probably 6 million molecules. Then if you narrow down to drug-like, but you can expand it using molecule that could be synthesized with few steps, like this kind of like enamine real type of library, you can reach billions. So you need to combine a QFEP with active learning and co-folding to really be efficient in the way you search your chemical space. And this is only taking account affinity. So the next step is try to use generative AI that is based on reverse diffusion in the binding side of the protein target. And we can have an affinity-based graph neural network trained on a QFEP, but then we can include other metrics that are relevant. So it could be ADME-Tox ML, that could be part of the reward function if you want. We can have also a synthetic accessibility score. So try to optimize this molecule using a parallel approach with a multi-parameter optimization strategy. And that is also taking advantage of all the data available experimentally from the client or from the literature. And then again, we want to be sure that we can link the conformation of the molecule bound to the target to the functional activity. And so the conformational ensemble could be very relevant to ensure you bind to the right conformation with the expected outcome. And then the final step is how do you bring into the game all other type of data that may be more challenging to include in the target product profile. So there could be some information from competitors, there could be information from the clinic, there could be omics data, maybe the client has access to, or maybe publicly available. And so using a knowledge graph solution kind of allows this different type of information to be included into your kind of decision process. And this at the end is really try to maximize the efficacy and the efficiency of the workflow. So the idea is to move faster toward the candidate nomination, and that means making fewer molecules per cycle and enable bigger jumps in the chemical space each time moving toward the target profile of interest.
All right, moving on, our next question. How can you transform current standard ML and physics-based solutions into an LQM strategy? How can it be integrated with the existing process and traditional approaches?
Yeah, that's another great question. So the idea is to try to start with the optimization of machine learning and physics-based method available to try to create this efficient workflow. So LQM, you can imagine, start from the tools that are available in the client, and we can expand on them. We can include additional data. For example, can we combine all publicly available data and the internal client data to create the best possible ADME-Tox machine learning model? And then on another side, we can take all the proprietary crystal structure or cryo-EM structure that are available in the client data set and use those to fine-tune the co-folding solution. So now we have kind of structural information, a unique customer LQM to predict the co-folded small molecule protein complex. We combine it with ADME-Tox that are optimized on client data, and we can use a generative AI to really try to link the two. So creating molecule in the binding site and this co-folded conformation, and try to maximize the affinity using maybe a PCM approach, and then try to maximize this ADME-Tox data. So all this LQM is based on data and tools that may be available on the client side, and we kind of create an optimal workflow, filling the gaps, ensuring is valuable in a prospective fashion. And we can use the knowledge graph on top of that to try to bring additional clinical information or competitor data or omics information that could be valuable. So at the end of the day, is really try to create the right infrastructure, combining AI, physics-based building blocks to solve all these challenges in one step and try to connect all the different pieces together. That's kind of what is the LQM at the end.
All right. Well, thank you very much for that one. When working with a partner to build an optimal LQM solution, how do you ensure data confidentiality, and how are client data used, or how client data are used, rather?
Right. So the LQM solution, it's important that it's optimized. If there are data available, we want to take full advantage of that. That is really the power. It's a kind of bespoke solution that is fine-tuned on the client or partner problem with their data available. We want to take advantage of everything we can, really. And But the way it's done is really specific for the collaboration, so there is no risk. And we have a very strong, quantum security group at SandboxAQ. So we really are able to firewall completely the collaboration, ensuring that all confidential data are safe.
Next question, how much does this apply to drug development for biologics?
So I can answer that one quickly. So basically, biologics are a very similar problem in a way to small molecules. It's just the physics are fundamentally different. So there are a number of companies that are out there right now that are essentially designing biologics very much in the same way that you'd think about small molecule developers. So I have on top of mind companies like Big Hat Biologics or Absci, both of whom are using essentially AI approaches to optimize the design of biologics towards this. This kind of an approach towards optimal target modulation. These kinds of approaches could be adapted towards an LQM approach as well. So certainly, while the main focus of Sandbox's effort in the past 2 years has been on the small molecule space, the general approach can be adapted to biologics, and is obviously a very fruitful direction to develop as well. I hope that answers your question.
All right, thank you. Sort of a comment and a question in one here. Surprised to see the main timeline and cost benefits at the clinical stage rather than discovery stage. Are there many timeline and cost benefits in the discovery stage?
Yes, I think what you're seeing is actually just the way the slide was presented. Actually, if you look at this, if you look at how the value gains happen, it happens primarily in the early stage. So both the target discovery and the compound discovery are the ones that have many factors of improvement. At later stages, it's mostly translated through from those factors of improvement. And you'll notice that the overall cost benefits decline as a factor of as a function of the accumulated cost benefit over time, simply because you get more dominated by other kinds of costs that are not as affected. So I would say, yes, the primary advantages are in the early stage, and these costs on the bottom are accounting for it cumulatively. And the main gains actually happen earlier on. Again, let me know if I've answered the question.
All right, referring back to how will this fit into the market, how can LQM help with operations, manufacturing, and process models?
Yeah, so that's a great question. So at the moment, we are focusing on discovery and development. Those are the next, I guess, steps. So in theory, potentially, you can imagine the key principles are still valid. So try to use all the information available and try to move from, something that is simple as memorizing everything online. If you think about like a large language model, right? It's kind of memorizing everything available online and then try to create a hybrid answer, mixing and matching kind of the information available. So the idea of the LQM is overall try to go deeper into the key principle. So for drug discovery, those key principles are understanding the physics and the chemistry. Can AI learn physics and chemistry? Because if it's able to understand those key basic principles, then the applicability domain will be massive, right? It doesn't anymore depend on the training set. So here, when you look at this other broader problem, I think the question to have something that is quantitative in the ability to predict, so LQM, will need to understand not just the data, but the key principle behind the challenges that the data are highlighting. So if you can bridge that gap, I think LQM will be applicable broadly on all those cases.
Okay, for our next question, what are the main caveats when integrating experimental data into LQMs, given that there could be errors in data or other confounding interactions like binding, allostery, and other knock-on effects?
Yeah, I think that's a key question. So the way we try to denoise experimental data is trying to include more physics and chemical understanding of the problem. So in theory, like if you're able to go from the experimental data to a full molecular understanding of the problem, you may be much more in control about understanding the error bars that are in the training set. So imagine you can control the level of theory of your quantum chemistry training set that you use for the machine learning force field, for example. You can benchmark and try really to understand what are the issue on the molecular dynamics side or your free energy perturbation. Is this converging? Is the conformation correct? So you're much more in control of the full understanding of your system. So when you rely more on this quantitative physics-based model, you may have a better ability to understand the limits and the risk. When you look at the experimental part, instead, I think there are a lot of unknown and potential kind of risk in the ability to assess how reliable are those data, right? And there are a lot of aspects, and especially if you start to move from simple, binding assay, you're already on phenotypic screening, the noise level is much higher. So the view is you may not be able to fully remove those errors. It's more like try to understand from a Bayesian viewpoint, what is the impact of that error? What is the confidence of your prediction? So you will be still able to make the best possible decision. However, you will be aware there is a risk. So there is a kind of explore, exploit type of strategy where you know that maybe that prediction may not fully reliable, but it looks really promising and you are happy to take the risk. So I don't think you can remove the error. It's more like you want to assess the error correctly and make your decision, understanding what is the risk related to that decision.
All right, moving along, do LQMs involve deep learning? And if so, what varieties are involved? Is it Gen AI? Does it use autoencoder architecture?
Right, yeah, so it's definitely, so the component behind that is taking advantage of the experimental data and the component that's taking advantage of all the physics-based simulation is deep learning. There are different types of strategies. I don't think LQM by definition use or prefer one strategy over the other one. And that is probably part of this bespoke strategy and solution. So the generative AI we're using, a sandbox, is a reverse diffusion in the binding side. That is kind of really what looks extremely promising and game-changing, but it's not a requirement. You can potentially create LQMs with behind the scene a wide variety of deep learning solutions. And these are moving and evolving extremely fast. So I feel like you can always try to include in your LQM the more exciting, the more innovative and game-changing type of technology. And that will power and make the prediction even more efficient and kind of exciting. But I don't think we want to focus only on one particular solution. And also it could depend on the type of problem. You may need to try different strategy. And often you have a retrospective kind of validation where you can benchmark and optimize your LQM for. And then when you feel like this is working and is promising, then you can deploy your LQM approach in a more productive fashion for prospective use.
Thank you. How are we using LQMs to also simulate drug metabolism in vivo before the drug matches the target site, and as a perfectly designed drug could fail metabolically and reduce bioavailability at the target site?
Right. So that's another great question. So in theory, if you think about drug metabolism, and we have some research, Sandbox, and also kind of in collaboration with NVIDIA, try to understand at a quantum level drug metabolism. And if you think about P450 enzyme stability, you can imagine as a multi-scale type of problem, right? So you can start from a quantum chemistry type of vision. I think in that particular space, you can try to model exactly the molecule, how is binding the enzyme active site, and then try to understand the reaction pathway. And we have like a very strong DMRG research line in collaboration with NVIDIA, try to push the boundary of quantum chemistry and quality of those quantum chemistry questions, especially when you have, heavy metal in the binding site. The quantum chemistry complexity is extremely challenging to model in a precise way. So the MRG is for sure very promising, and then try to bridge the MRG to other type of solutions that are more like the normal strategy, and then bring that to maybe a binding affinity prediction, because there is a specific orientation of the molecule that is required for the reaction to happen. Can you learn from that? Can you modify the molecule to avoid that position such a way the reaction barrier is higher, and so the molecule has a longer half-life? And then you can bring that to a larger scale and maybe bring in experimental data. Again, going back to the LQM vision of molecular understanding, but then bring all the data available, and depending on the client collaboration, there could be quite a lot of microsomal stability or hepatocyte stability data, and we can bring, literature data as well. And so again, try to have an LQM that can jump these different levels of details and provide an effective prediction for every single molecule to help medicinal chemistry design that is really taking advantage of everything available in hand.
All right. Are you leveraging single-cell genomic expression data? And if so, where are the data sourced from?
So it depends on the collaboration. So we have a collaboration where the client is extremely capable to provide game-changing omics data, and we are able to include those. There are other collaborations where genomics can be important, especially for target identification. The way we could try to include that information, it really depends on the type of question we are trying to solve and the type of data and the quality of data that the client can provide. We can potentially access publicly available data, but very often when there is the specific need, the client has some unique kind of edge that we want to ensure is able to take full advantage of. So the vision is the data from the client are often extremely exciting, game-changing, and the question is, how can we ensure we are using them in the best possible way? And so there is this close collaboration between SandboxAQ and the client to predict and create the best possible LQM that can take full advantage of that. In one case in particular, we're looking at target identification. How can we use novel genomics data for that? It's not exactly what you are describing, but I think in theory, the view would be we should collaborate together in a constructive way to try to design the right solution, the right LQM to really exploit those information.
All right. Well, we are coming to the end of our time here today, and so this will be our last question. If we weren't able to get to your question, we'll try and get back to you personally after the webinar. So to wrap things up today, what is the best way to deliver LQM to optimize researcher workflow?
So in our view, the best way is to kind of, first of all, create this open and constructive collaboration between the client that is really an expert in all the available data, the computational chemist or machine learning expert in the client side that is already kind of try different ways to exploit this information. And so they already identify the challenges, they already identify the bottlenecks, they already know what didn't work. So, and there could be already a definition of success that is clearly defined. And maybe there are some data we could use as a retrospective validation test. So in that particular scenario, the idea is the LQM should expand everything that is available and be really optimized based on the feedback from the client. So it's like almost we're expanding the team from the partner side. We're bringing AI expertise, computational chemistry expertise, quantum chemistry, physics-based method, but also cloud and software engineers expertise that could help to kind of probe different strategies and solutions. We try to combine these tools. We have like a wide range of tools and building blocks. We try to combine together based on the feedback also from the partner. And then we tested in a kind of cycle on this retrospective set. And only when we reach that target on the retrospective set that we kind of prove the value. We prove this is working. It is proven to, at least retrospectively, to solve the problem in hand. Then with the kind of agreement with the client, we move toward a prospective approach and deployment. And so that is really where, again, there is a real success definition. And that is where, and that is experimental, right? So we will use the LQM, design together, optimize together, and then we will have to prove it's working in a prospective fashion, experimentally, providing prediction that are in agreement. You know, it could be a new hit identification, a new lead molecule, or it could be an ability to identify the right target for this particular molecule that before was unknown, maybe from phenotypic screening. Or it could be identification of a new biomarker in clinical development. So this LQM needs to be like a collaborative optimization between the partner and Sandbox or the expert that will provide that service.
All right, well, thank you so much to both of you, and thank you to everyone for joining today's webinar. As a reminder, this webinar will be available on demand within 24 hours. We appreciate your participation, and we look forward to seeing you at future events.
Thank you.